please welcome our speaker.
Yeah, thank you very much, Grace, for the very nice welcome, for the very nice introduction. I must say one thing, I have a really nasty cold, so I hope my voice lasts. I've got a lot of water, but someone might sort of, have to go and get some water during the talk, but I try to sort of speak loudly enough. I guess you can all hear me in the back. Awesome. So I saw a few of you already today in the library when I was preparing my talk. It's great that you all came to my presentation here. Yeah, as Grace said, my name is Michael. I'm a professor for evolutionary anthropology at the University of Zurich, but I've been working on dolphins pretty much, all my scientific life. Did my PhD at the University of New South Wales in Sydney in Australia. Then a postdoc, and I was very lucky to get to become part of this long-term research in Australia. You might ask yourself, anthropology, how does that fit with dolphins? I hope this will become clear during the talk. I also worked on orangutans quite a bit in Indonesia, but dolphins are my first love, and the marine realm is my first love. So I really, put most of the effort I do into this amazing work in Shark Bay that I'm very sort of glad to be able to be part of. And one of the major aspects of that work is really sort of, looking at social complexity in these Indo-Pacific bottlenose dolphins. You've got killer whales right here in front of you. You probably heard all about the social complexity in killer whales. I would say that our dolphins surpass the killer whales quite a bit by that. And also when I talk to primatologists, everyone says, that usually the primates, the chimpanzees have very complex social structures. But I hope that by the end of the talk, you will see that also these very complex social structures have evolved independently, in the marine realm. Remember, the most recent common ancestor of us, of the primate lineage and the marine mammal lineage, lived about 120 million years ago. Independent evolution, yet very similar outcomes over time. And I will sort of talk to you today about our findings that long-term research really was able to, uncover. So in anthropology, of course, we are interested in the human condition. Why are we the way we are? And that is sort of a result of multiple adaptations in the hominin lineage. But of course, in modern anthropology, really, we do a lot of comparative work. So, in the past, anthropologists, they looked more as humans and very little sort of, outside humans to primates or even further away. And modern sort of, anthropologists, they don't look only at our species, but also beyond that. And the idea is really to look at the similarities. And once we've sort of carved out the similarities, we can then look at human uniqueness. So basically, once we have these similarities between our own species and other taxa, so for instance, we have a very long or very slow life history. We've got these ridiculously large brains as humans. We have culture and tool use, and we find, very sort of, high-level cooperation and very complex social systems. And this is really what describes sort of human species, but actually by looking more into other taxa, we find exactly the same in primates and also in marine mammals. You see here one of the dolphins in Shark Bay. It's just coming to the surface, so this is, exhaling, and this dolphin is carrying a sponge, and they use these sponges as marine tools or as tools. And I will, if I have time, talk about this at the end of the talk as well. So the idea is, of course, once we sort of, map the, and there should be similarities, we can then, focus on human uniqueness. So many of these traits that I sort of talk about here, life history, brain size, social complexity, culture, tool use, cooperation, we find at least in a basic level in many other taxa. And once we sort of, look at the similarities, the evolutionary foundation of those, we can look at human uniqueness. So, but of course, that will, also pose the question, what actually counts as complex, what is actually a complex society? And there have been, throughout the time, different, approaches of how to define this, starting, with Hind in 1976, looking at, different relationships within these populations at a hierarchical level. Kappeler and Van Schaik, they basically based this around structural and organizational complexity. This could be a lecture by itself. and Krützen and colleagues very recently, they looked at sort of, multi-level and hierarchical nesting of, of individuals within these populations. So you start at the individual sort of, might form a smaller group at the first hierarchical level, then these sort of smaller groups get together at a second hierarchical level, and so on and so forth. So basically, we've got these several levels, and that might describe a complex society. And if we apply this framework here by Krützen and colleagues to vertebra, we can actually see that these multi-level and hierarchical population structures are found, first of all, of course, in our own lineage, in the primates here. They've been very well described in the primates. We also find these in elephants. And you can see here the sort of the second large cluster is actually that of the toothed whales, of the marine mammals. So social complexity is also found very frequently in very different marine mammal species. And as an add-on here, because this will be important for the rest of my talk as well, we also find that in these taxa here, if we basically look at the 3 peaks of mammalian brain evolution, we have that in primates, we have this in elephants, and we have this in cetaceans, that at least in cetaceans and in primates, animals cooperate at very sort of multiple and nested level. And this sort of kind of cooperation that we find in this taxa we can describe as very complex. So the similarities in these taxa here, and remember, they are sort of all independently involved in primates, elephants, and cetaceans. We've got the very large neocortex size when it comes to brains, very complex social structure. Social learning occurs, or what we call culture, so that basically information is transmitted between individuals within generations or between generations via social learning. And we've got these complex fission-fusion societies, and I will sort of explain in a second what this means. We also have in some of these taxa alliance formation, and alliance formation are recurring coalitions among the same individuals. That was a definition by Harcourt in 1992. And what Chappé then said in 1995 is that this alliance behavior, and this will become really important for the dolphins, lies at the heart of social bonding and cognition. The ability to form alliances generates the most complex social structures in the animal world. And what we found out in these bottlenose dolphins in Shark Bay, that they form alliances on several levels, when it comes to basically, reproduction, and I will explain this in detail. So these alliances are actually very rare in the animal kingdom. We find them, very common in humans, chimpanzees, bottlenose dolphins, in some monkey species, and very few other taxa. But alliances, these groups, basically recurring coalitions when actually animals work together on something among the same individuals, is pretty rare in the animal kingdom. And this brings me to our study in Shark Bay, which is about 850 kilometers north of Perth. So we've got the Australian continent here. Perth is, in Western Australia here. Shark Bay, roughly 850 kilometers to the north. Shark Bay, as it's in English, or Gutharaguda in the local Aboriginal language. Shark Bay is roughly the size or half the size of Switzerland, which would be a little bit bigger than, Vancouver Island. And it's divided, in the Eastern Gulf here and in the Western Gulf over here. We work out of Monkey Maya in Shark Bay. Has anyone been to Monkey Maya by any chance within the audience? My kids have been, of course, but no one else yet. I can really recommend this. And, since 2007, we work out of the Western Gulf as well, a place called Eustace Loop. And of course, it allows us, to compare animals in these sort of two gulfs here. And Shark Bay is a UNESCO World Heritage Site, so it's got the largest and the most diverse seagrass beds in the entire world. There are many stromatolites found, these beds, stromatolite beds within Shark Bay, and it also has the largest dugong population in the entire world. And for this, Shark Bay has become a UNESCO World Heritage Site quite a while ago because of its outstanding natural values there. But this was not the reason why my colleague Richard Connor actually went there in 1982. So he had heard somewhere in National Geographic that in Australia, there were dolphins coming to the beach, and one could feed these dolphins at the beach. And he wanted to study dolphins for his PhD. That was his lifelong dream. It's still his dream. And he's been working there. So he and Rachel Smolker went there in 1982. They hitchhiked up north, and they actually found straight away that there were animals right at the beach. And they saw these animals day after day after day. They seemed to be not shy, so they were used to boats. So they realized the potential. And then in 1984, they basically started a study where they systematically started to take behavioral data of these dolphins. And this has turned into the second longest running study of any marine mammal in the world. The longest one is in Sarasota Bay in Florida. So we can actually claim, the second spot. But I would still argue that actually most of what we know about small cetacean behavior, inshore Odontocetes, these small cetaceans, we know from Shark Bay. And I'll tell you in a second why that is the case. And I came on board in 1996, so in the last millennium. That shows my age a little bit as well. When I was starting my PhD, with genetic analyses. So we started to take, biopsy samples of these dolphins to do genetics. And then in 2006, my colleague Stephanie King from Bristol University joined us, and she sort of, brought in a lot of acoustics and behavioral expertise as well. So, and how do we get the behavioral data from these dolphins? We are very, very spoiled because the animals are right there. So we go basically when the weather is fine. On a day like this, we look out of the window. Our views aren't as nice as these ones here. But we go to the water's edge, it's flat, we jump on the boats, we've got small boats, and we are out there, and the animals are right there. Okay, so we don't have to go very fast, and Shark Bay, it's a huge body of water, but it's very shallow. So we call it a duck pond, really. It's not ocean, sort of not the big ocean. It's fairly, safe and easy to work there. But the main tool we use is photo identification. So we basically have these cameras here with sort of these long 400mm lenses, and we take photos of the dorsal fin. And here you can see these three guys here, very easy to identify these dolphins just by the look of their dorsal fin. And each dorsal fin is unique. It's like a fingerprint. So we basically have identified more than 3,000 animals, over the past 40 years. So we do these behavioral surveys. So we want to sort of, find out who is there, so we can identify the animals, by their dorsal fin. And good people, I was never good at this, I'm too short-sighted, but good people, they do this in situ. They identify the animals on the water. And all of these animals have names, 3-letter codes. So it's much easier to remember, spot and real notch and high and bottom hook compared to A57, and H59, like it is with the killer whales. We find out sort of, where they are using GPS. Before that, there was triangulation with nearest landmarks. We threw out away all of this data because they were not accurate enough. Now with GPS, we know exactly where they are. And of course, we sort of, want to find out what they are doing. So are they traveling? Are they socializing? Are they foraging? Or, what kind of behavior they actually show. And as you can see here, this is fairly cool. The animals are very close to the vessel. The water is pretty calm. This was Richard here in the 80s, so the fashion was very different there, as you can see. But, the dolphins are very close. They're very habituated to what we're doing. And that really allows us to look at this, into more detail now. Sometimes we also do individual follows where we just pick one individual and we follow this individual for a whole day. And this is, of course, a very different kind of data we get rather than these surveys, which are, short snapshots, who is with whom and where. The individual follows, we can then really sort of see, for one individual, who does this individual associate with, does it travel afar, and so on and so forth. And the conditions allow us to do this in a very sort of, straightforward and easy manner. As I said, since 1992, more than 3,000 dolphins have been identified. We have a catalog, and we have demographic data for each of these animals from birth to death. Because I think this is really important. Dolphins are born, they live and die in the study area. Dolphins, come up to 40 years of age. That means we can follow these animals from birth to death. And this is sort of, what no one else really in the world can do. We are very lucky in that regard. Shark Bay is such a unique and, rich habitat that the animals don't have to travel very far to follow their prey. The prey is there all year round. And that, of course, is really sort of, good for what we are doing. So taking these genetic samples, that is, when I started there with my PhD, that's me with a sort of, it's a modified 22 caliber rifle here. And this is not inches down here, this is only centimeters. So we've got these very small biopsy darts, and we basically shoot the darts. Here you can see slice and dice, and we shoot the dart, and that was a good hit below the dorsal fin. The dart just bounces off, takes a very tiny sample, here of skin, and we use this, for DNA, to take DNA. We also use this for foraging ecology, looking at stable isotopes and fatty acids analyses. So we can use this kind of sample, for multiple different kind of analyses. And people are really worried about this. When I did my PhD, I was, standing there on the bow with a gun pointing down the dolphins. People saw this, from the beach in Australia. They called the water police. The water police came to us, and they want to know what we were doing. And I was saying with this German accent, yeah, we are just darting the dolphins. Yeah, and so they weren't too happy. But of course, we had all the permits, and then we didn't, weren't sent into jail. But whoops, this is actually working pretty well. And you can see here my student Svenja showing this is sort of one of the dolphins coming up. And I might just go back a little bit. It was a little fast. I didn't expect this to happen so fast. So basically, we've got these small boats here, and then people, this is Timon taking photos. This is Svenja with the gun here. Animals are really relaxed around the boat. And then basically she takes aim, that animal comes up. Bang, there's a shot. Animal stuff, goes away. And then basically the darts float at the surface, and we take the biopsy samples. And of course, the big problem was for us, we were really worried that animals would, just start avoiding us. They would just see, oh God, these humans, with a biopsy gun, and, for a behavioral project, that would be the dead, of course, the end of the project. But that didn't happen. So the animals, as smart as dolphins are, they don't seem to associate it, with us humans. And we've taken more than 1,000 samples, and the animals, are still alive, and they still come to the boats. And we are really sure there are no long-term effects, but we can get so much out of these samples that I will talk about in a second. So what have we found out from the behavioral perspective? I will focus mainly on the male stuff today, on the male behavior today. That doesn't mean that the females aren't as interesting, but Richard and our team, we mainly focused on the males and their mating behavior over the years. So what they found out is that these sort of males, they have very strong associations during the year. So they basically see always these pairs or trios of males together throughout the entire year. Remember, we can look at these, or we can identify the animals very well by looking at their dorsal fin. And what seemed to be the case is that these pairs and these trios cooperate to monopolize females that are in estrus, that are cycling, okay? So what these males do, they basically separate the female from the group. And what's really important in shark bay dolphins is it's a fission fusion society. So you probably hear about the killer whales, where we have these pods that are sort of really defined, the pod size, and the members of the pods are defined. This is not what we see in shark bay. Shark bay is more fluent. Groups form, groups sort of split up, groups, new groups might form with new members, and then sort of, and this might happen. So it's a very fluent system. That's why we call it a fission fusion society. This is how we are. We now basically form one group. If someone came from Mars today, they would say, okay, these guys probably are one human group, but tomorrow we would find the members of this group in different subgroups again. So groups form and basically split, and this is what these dolphins do as well. So they take the female from all these groups, and then they might take the female out of the home range, and they monopolize the females for a very long time. And what they all do, you can see this here very nicely. We've got these 3 males and the 1 female. And during this, up to 60 days, they all mate with the female. There's an aggressive defense. So females are a very rare resource, if I may say this. In biology, we say it's a resource, because obviously for the males, they want to mate with the female. But at any time in the population, there are only very few females that are ready to mate, because this is what we call a biased operational sex ratio, because most females are either too young or they have calves that they are nursing, so they're not ready to mate. So we've got many males who are ready to mate and very few females. And of course, this is why there's a lot of fight over access to these females. So they aggressively defend. You know, some alliances, you know, might want to sort of try to steal a female, so there's an aggressive defense. And as I said before, there's a frequent matings of all the males with the female. What is important, you know, in primatology, it's actually very easy. You are on land, you can usually see, you know, very easily just with binoculars who is mating with whom. So who has access to the females. You might remember chimpanzees, you know, when the chimpanzees females have the swellings and the sort of alpha male is just behind her. So it's pretty easy to observe who's actually the lucky one. With the dolphins, we just don't know. Because first of all, you know, it's very hard from the boat to see whether an animal is male or female. Killer whales have this massive sexual size dimorphism. We don't have that in the dolphins. They are pretty much the same size. Sometimes they turn around and we can see, you know, the anogenital area, then we can see it's male or female. Sometimes we see an erection, well, then it's pretty obvious, you know, what we have there. But actually, who has access to these females of these three guys here and who is successful is very hard to see. We fly drones now over them. It's a little bit better. It's still tough. But with genetics, actually, we can actually decide, who is actually the father by having genetic samples, okay? So, and also, you know, they've got synchronous displays and vocalizations around the female to strengthen the bond, you know, among the males. So it's a really elaborate behavior on this, what we call the first order alliance level, okay? So these pairs or trios are first order alliances. And that was, you know, very cool. You know, people, and this is sort of, you know, what it also looks like here. So we've got one female up there, and you see the three males here converging behind her, and they make sure the female stays with them for up to 60 days, okay? You can see here the female is going down, long breath, she's going down, and the males know exactly where she is. Sometimes, you know, we have it that these two males would follow her while the other guys are sort of going off foraging, and they take turns, but they really make sure the female stays with them. And they threaten the female. If the female wants to leave, she gets sort of physically, you know, she can get physical violence against her, but also some popping sounds, you know, where they actually partly open the blowhole and goes pop, 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 pop. And apparently that really makes the female stay. Doesn't work in humans, I've tried that, you know, but for the dolphins, it's a very strong signal, you know, very strong signal. Okay, so, and all the males, as I said, mate with the female, and of course, that is interesting from an evolutionary point of view. You know, we've got one female, one male, and the female might have a calf every 2 to 3 years, and 3 potential males. You see the problem already, don't you? Yeah. So who is the father? Yeah. And this is something we sort of, you know, wanted to understand, and I will come to that in a second. So what we have is this really complex formation in these alliances, and this is what Richard found out in the 1980s. Okay, we've got these pairs or trios, these so-called first-order alliances. So you've got real, notch, and high, trips, bites, and cetus here, and I can't remember. I think this is bottom hook. I can't remember the names of these guys, but you know, this is what we call a sociogram, where basically these lines indicate that these guys spend a lot of time together. Yeah, first order. So there's cooperation on this first order level when it comes to mating with females. Really cool, not described anywhere before. In the 80s, people were really excited, but this is nothing of what is going to come, you know, and the discoveries after that. So when Richard extended the research area in 1994, what he actually found is a different group. So we've got a group of 14 males here, which is, you know, he called a second order alliance or a super alliance. And this group here is very stable as well. And you can see that within this group, you know, for instance, hobo here, they form first order alliances, you know, sometimes with myrtle, with wow, with a gripe, and with lech here. So basically, out of this group of 14, smaller subgroups, you know, are being formed of 2 to 3 individuals, okay? And these sort of 2 or 3 individuals, they then basically are together with the female again. So they do this herding with the females. So the pairs or trios are always with the female, but here you've got a larger male group, 14 individuals, and you know, at some stage you might see, you know, hobo, lech, and gripe doing the herding with the female. At another instance, you might just see lech and pig and wow doing the herding of the females. So they basically recruit, always these pair trios are getting recruited out of this larger group of males here, okay? And this is sort of, you know, what Richard then found is really what we have here is this nested alliance structure, okay? And this is what he called a second order alliance. And of course, this was then, getting really interesting because the question is still the same. We've got all these males cooperating even on different levels, but we still don't know how this translates to paternity and, paternity data. So, and basically what happens is we have this very complex cooperation and strong competition over access to an extremely rare resource, the fertile females. We've got basically here two second-order alliances engaging into fights. So it looks like, head-to-head, we know something is happening, and then bang, they start fighting each other. And one guy here, see down here, he's just, running away. That's the best he could do, so they don't really engage in these fights. I mean, we find that animals are getting injured, they don't kill each other, but basically there is a lot of fighting over access to females. So it's not the friendly dolphins that smile all the time that people know, I want to live in a dolphin society. I don't want to live in a dolphin society. It's really, really rough and tumble what's going on there. So these two second-order alliances, they engage into fights over the females. And of course, that is a behavior that us behavioral biologists or evolutionary biologists, we are really fascinated by this. So why do the males cooperate to obtain a resource that cannot be shared? Remember, one female, one offspring, but several males that cooperate to mate with the female. But only one of these three guys can be a father. So why do the other two dolphins help the one dolphin to be a father? It doesn't make sense from an evolutionary point of view. We are here to pass our own genes into the next generation. Unless we cooperate with dolphins with whom we are related, okay? Because, my half-brother shares 25% of the genes with me. My brother shares 50% of his genes with me. And that means, if my brother is successful and, that basically, I would not be successful, that means that obviously that I get, a fitness benefit, even sort of, because my brother passes on, 50% of the genes that we have, it's very likely that some of the genes that I have are being passed on by my brother when my brother is successful as well, okay? So a simple mechanism of cooperation is kin selection, okay? Blood's thicker than water, you cooperate with kin. Basically, from an evolutionary point of view, not a problem at all because kin has very similar genes to yourself. More complex mechanisms are reciprocity or pseudo-reciprocity. This is more kind of, I help a tit for tat. I help you now, you help me later. But of course, that requires some scorekeeping, yeah? So because you need to know, you help me now, I can trust you, but you owe me basically a favor sometime in the future. And that has been linked to large brains and to cognition because you need to sort of be able to keep track of all of these relationships. So, and this is what we find in humans, and this has been proposed for the dolphins as well. And of course, the question was sort of, what are the fitness consequences, of this cooperation? Is it that some males are super successful? Is it that paternities are shared, equally? And of course, this is what we can do with genetics. This is where we can really easily use genetics and look at paternity success in these dolphins, even if we can't really in detail, see who is successful when it comes to the actual mating. So when I finished my PhD, that was one of my first papers here. I basically thought I had it solved, and everything is fine, and we can move on. But of course, as you know, science is very different. Just by answering one question, you actually pose another few more questions. So what we found is that kin selection here seemed to explain, these small trios and pairs that we see here. So all of these guys, when we looked at this, they were more closely related to each other within these alliances than compared to the population average. So it seems that kinship seemed to play a role there. But in the same population, when we looked at the super alliance here, at that larger group of males, so we actually obtained samples from all of these individuals and we then did relatedness analyses, we actually found that kinship does not play a role here. So maybe this is based, on reciprocity or something else. We didn't know it. So, but the point is that we claimed then, there were two contrasting strategies within the same population, okay? Some of the animals are related, others are not. And that was, a good paper at the time, and we thought we have solved it. And then, of course, as it is, we increased our sample size. We actually went further out. We got more data. We got more long-term data. And then we actually found that the story is much cooler and much more complicated than we actually originally thought it was going to be. So these second order alliances, these large alliances that I just talked about, they are actually the norm in Shark Bay. And this is the core social unit. These alliances are formed when the animals, the males, are between 10 and 12 years of age, and they do not change after that. So you're stuck with your second order alliance partner for the rest of your life. And of course, that determines, whether you're successful or not in fathering offspring. If you sort of end up with some duds in your second order alliance, you might have problems, So it's very important that you choose the right alliance partners. So relatedness really does not explain cooperation. It seems that relatedness does not play a role, so that easy explanation for cooperation in dolphins is not valid. We have to evoke more complex mechanisms that might explain this. We also find, so if we look at the different alliances, this is just, an aerial view of the peninsula. Here's Monkey Maya, our base, and this is sort of, going up north, distance in kilometers. We find these different alliances, at different positions. They are fairly sort of, easy to find, always in the same area. We find, that these contract rates vary between second order alliances. So some order alliances are really good. They are with females all the time. We see them with females all the time. Others are not as good. Okay, so again, really important. You want to end up, with a good alliance that, has females all the time from an evolutionary point of view. We also find that these paternities are equally distributed among alliance members. So also a sign that these alliances are not based on kinship. You might remember lions, when lions basically take over a pride, they kill all the offspring. So as infanticide, the females start cycling again. And then, in some of these groups with alliance, we have a case where one of the males achieves all the paternities while the other guys don't get any. And of course, that begs the question, so why do they actually cooperate? Why is this stable? I would just basically, want to reproduce myself. But in the lions, these alliances are based on litter mates. They are all related to each other. So that makes sense. If one guy gets all the paternities, because of inclusive fitness, I don't really care because, he's my brother. Yeah, so in lions, it explains this. So in dolphins, with the genetics, we found that the paternities are equally distributed among the alliance members. But actually it gets a little bit more complicated, that actually second order alliances get even together to so-called third order alliances, which are strategically cultivated, and they produce this huge open network here. So this is a network, and some of the lines we can't unfortunately see because of the beamer there, but basically all of these males are connected in a single network. This is, Monkey Maya here again. This is the water. This is roughly where these dolphins occur. These alliances, each dolphin is a single individual. You can see the thickest lines are, within these alliances, but they always have relationship to other alliances as well. So these third order alliances are strategically cultivated, and they produce the largest open, fission fusion network that has been documented outside humans. So dolphins are very special in that regard. And what's also important is that, within this large alliance network, animals have very differentiated relationships among non-related individuals. So kinship does not play a role in explaining what we see there. What we find, though, is, with our 40 years of research, is that the strength of these third order bonds in the third order lines also predict access to females. So again, this third level is also to some extent linked to access to females. And when we look at consortship rate here, or maximum consortship duration of days, so there's always a measure sort of, how often these males have a consortship, how long are these consortships. Within alliances, we can see there's a positive correlation between consortship rates and within alliance strength, both for, consortship rate and maximum consortship duration in days, but also between alliance strengths is also positively correlated here. So this third order level also is functional and is linked to reproductive success. So that is, describing these alliances. But of course, what is really important, and also the long-term data allow us, is to look, how do these alliances actually develop over time? Okay, so remember, animals are weaned when they're 2 to 3 years of age. So they're away from mom. And then these alliances, they start forming these alliances when they're 12 to 13 years of age. So we've got this 10-year gap in between. And the animals must do something in that time, and this is really sort of, trying to get the best alliance mates. So we have this large gap between weaning and sexual maturity and alliance formation. And here we have, 59 males with 13 years of detailed behavioral data on each male. So this is quite sort of, a feat to get this, really close data. And what we sort of, what my student Livia found is that these social relationships during adolescence, they predict alliance formation. So the guys you hang out with when you're very young, that predicts who is going to be your alliance partner, your alliance buddy in the future. So it seems age is sort of very important. It kind of makes sense intuitively because, you've got the same age cohort, you grow up together, and then, you form these alliances as well. We find that these social networks during adolescence are to some extent explained by relatedness. So you hang out with individuals to whom you are related, okay? But interestingly, once you become adults and this changes, then basically adulthood here does not really explain this anymore. And then we were also wondering, maybe if I can say this quickly, because people always criticized us. They said, right, you say it's not based on relatedness. But what if, for some demographic reason of the population, there are no related individuals available at the time when these dolphins form these alliances, then they can't even choose. So we asked that question, when these animals are young and when they grow up, do they have related individuals around with whom they theoretically could form these alliances? And this is sort of, what we looked at here, with all of these different males from these different second order alliances. And there were lots of individuals around at the time to whom they were highly related, yet they did not choose to form alliances with these guys. Okay, so that argument we can dismiss. There are lots of related individuals around, but they choose to actually use other individuals to form these alliances. So kinship, the basic explanation for cooperation in dolphins, really does not seem to explain this highly cooperative behavior. What we could also find is with the genetic data is, these well-integrated popular males within these alliances and, with very strong social bonds to other males, they seem to sire more offspring. So if you're popular, if you're well integrated with your mates, that seems to have a positive influence on your reproductive success. And so it seems really that investing in these social bonds is adaptive as it is directly linked to fitness, to the number of offspring you can produce. And of course, it makes intuitively sense. We've got these animals with these really large brains. Large brains are extremely costly. They need to be maintained, and they need to be there for something. If they did not use their brains, it would be sort of, basically hard to explain why they have them in the first place. So it seems that, this social life, investing in these social bonds is really important in these marine mammals. So this is what we found over the years within Shark Bay. But as an evolutionary biologist, of course, we are really interested in sort of the evolution of this sort of complex societies, And if you look at Shark Bay, this is really sample size 1, isn't it? We've got one population where we find this, and we still don't really know sort of what caused the evolution of this complex, of these complex alliances and the cooperation there. So Shark Bay is one data point, and the previous work so far has been mostly on these proximate mechanisms of how these alliances are built, how they are maintained, in terms of reproductive success, how this translates to reproductive success. But I think it would be really interesting to look at the ecological drivers of social complexity in several populations. Okay, so that we basically not look at one population of dolphins in Shark Bay, but look at several populations, and then basically ask the questions: what are the ecological differences, mainly between the sites, that might explain differences in social complexity or in alliance formations of these dolphins? Of course, it's always a great idea, easier said than done. The question is here, are there any populations nearby and accessible to researchers? Are these populations genetically similar to each other or not? Because if they were not genetically similar to each other, People might say, well, this is a confounding factor. All the differences that you see are based on genetics and not, on the environment. And is it also possible to characterize and quantify habitat and social complexity in a timely manner? I think, working on marine mammals is tough. It takes a long time. So, is it possible to replicate to some extent what we do in Shark Bay, in a much shorter time frame? And the answer is, I would say, yes, I think in Western Australia we can do this. So we have access to several well-studied populations. This is Shark Bay here. There are sort of, well-studied populations down south around Perth. We've got, populations up north here. What is really interesting is, so we've got these, several populations along the coastline, and they actually found in sort of several bioregions. So each color here represents a different bioregion. We've got tropical waters, in the north of Australia, temperate, subtropical waters here, and warm to temperate waters in the southern, of Western Australia. So this is going to be important. Some of these populations also have some long-term behavioral data. So we find that, in some of these populations, there seems to be a lines formation, but not in others. So there is some behavioral variation that we already can document there. There is sort of, also variation in annual abundance of these animals among these sites, variation in predation rates among these sites here, and the mating seasons also differ and they happen at different times. So there is some variation among these populations that, of course, this is what we need. We need variation in order to test something. If it was all exactly identical, we couldn't really do any tests there. So there seems to be behavioral variation among the different sites. And also what sort of, we found with genomics work and genetics work, this is work by my PhD students Sam Whitwer and Svenja Marford, is that these bioregions or bioregions seem to drive genetic diversity. So we got genetic samples from all these different populations here in Western Australia, and they really cluster according to bioregions. So basically all the populations up north here, this is sort of, one genetic cluster here. This, a principal component analyses. We've got the populations from down south, they cluster here. They're genetically more similar to each other than to the other ones. And we've got the Shark Bay dolphins over here. What we have within these bioregions is so-called isolation by distance patterns. That means, that the further you go away from one population, the more slightly these change genetically, but there is gene flow within populations within these bioregions. Shark Bay is somewhat different, which I think is pretty interesting, given that it's sort of right between in the middle here. I think we know the case, and I will talk about in a second why this is the case. But anyway, we were a little bit sort of, surprised to see how different it really is here. We also find, looking at heterozygosity from north to south, so how, what the genetic variation is, that sort of is decreasing from north to south. That is sort of, in line with a scenario where dolphins actually started to populate the coastline here from the north and then went further south, so it's, these always, forming new populations, new populations, new populations, and new populations. And doing these series of founder events, you lose, of course, genetic diversity a little bit. And doing some demographic modeling, that was my postdoc Adrian, and Svenja, and then Sam again, we basically find that the source population is in northern Australia, and then basically Tursiops aduncus, the population went down the coast, after the last glacial maximum. Shark Bay was only founded 12 to 13,000 years ago, because that was really when the bay was flooded, so it's very young. And basically genetically we can find this. What we don't know is whether Shark Bay, was actually then founded from a split from the northern population or via admixture from the southern and the northern population. So that's something we're still working on at the main point. But the main point is here that these dolphin populations are of the same genetic stock. So the differences in social complexity seems less likely based on genetics. So we can not dismiss genetics, but we can see, it's a very recent history. Although we find differences among these dolphin populations genetically, given that we use genomics data, we find even the tiniest differences, they will be significant. We've got basically a very sort of, large, sample size, and basically because of that, even the tiniest statistical differences might become different. Okay, so we've got a lot of gene flow within these bioregions, but very little in between. So the dispersal patterns also differ among these populations. Okay, so we know the genetics of these populations. Remember, the idea is to extend the study that we have beyond Shark Bay to understand what are the ecological drivers of social complexity. And I think this is still a very relevant and important question. So the common ecological levers, you look at is basically food distribution, has been sort of, linked to animal, complexity, predation, habitat openness. That's one thing. Predation is a big point. Temporal environmental uncertainty also sort of, has been linked to group formation and social complexity. And, fission fusion dynamics, as we see in these Shark Bay dolphins. Remember, we talked about fission fusion dynamics. They decouple the social group from the foraging party, and basically that allows groups to track spatiotemporal variation in food. So it's all of these sort of different questions, or potential models that could be invoked to basically, explain social complexity. So it all seems to be about sex, food, predation. And the problem is, of course, here, how do we measure this very quickly and very briefly? Shark Bay, we are there for 40 years. So I could say, okay, we just repeat everything, in all of these other populations for 40 years. But by that time, I'll be long dead. Most of you might be dead. And of course, this is not really satisfying. So we need to come up with some measures or some methods that allow us to do that. And I think one of these things that I think is very exciting is an environmental DNA. I heard some of the instructors here is working on environmental DNA. Is she here today? No? Bummer. Okay, would have loved to have talked about her. But basically, this is really something that allows us to at least look at some aspects of environmental differences, very quickly. So this is what my PhD student Manuela Bizzozzolo did for her PhD. So we wanted to basically find out, how do these different areas where our dolphins occur in Shark Bay, how do they differ in terms of what's in the water? So of course you can go in the water, you can do trawling, you can do fishing and all this stuff, highly, it takes a lot of time, might not be really representative. It's, very time consuming, very costly as well. You probably have all found out, running all these boats here, is a lot of effort. But I think eDNA, you just go out there, you basically sample water, you run it over a filter, you take the filter, you preserve whatever is on there, the DNA, of any organism that was in the water, and then you take that filter, you take it to the lab, no CITES, nothing is required, take it to the lab and analyze it there, and you get a very good idea of actually what's in the water. And remember, what are these dolphins after? It's food that explains, social complexity. It's basically predation. So we can also look at predators of these dolphins. And in Shark Bay, for instance, we have lots of predators, big tiger sharks, bull sharks. There are no white pointers. They are further south in Australia. 90% of these dolphins have shark scars. So we can actually see, do the dolphins, try to avoid these sharks, looking at the landscape of fear and so on and so forth. So what Manuela did, basically, this is a Peron Peninsula. Here's the Eastern Gulf, Western Gulf, very quickly. She took water samples on all of these different sites here. Okay, so each green dot is basically a site where she took the water samples. And what she found is a lot of sort of, fish species, on a genus level, family level as well. But she also actually discovered another 72 genera that were not documented for Shark Bay based on normal methodology. She also, and this is as a side project, actually looked at lots of tropical taxa that were not even supposed to be in Shark Bay, but because of the water warming, I will talk about this at the end of my talk, she actually found that lots of tropical taxa also now seem to be within Shark Bay. And looking at this, eDNA and the conventional surveys, we can find that usually eDNA and conventional surveys, they overlap fairly okay, but usually eDNA is much more sort of bigger. We always have more taxa in eDNA than in conventional surveys. So what Manuela then did basically, so she used the genus richness here at each point that she could, each sampling point that she had, and she modeled that against satellite habitat variables and then extrapolated it to unsampled water. So basically, she could find, the richness of fish. So where's, a high richness, where's lots of diversity in our sampling area here. So basically, this is a map, that was based on her point sampling here in the Western Gulf as well. So this is the Eastern Gulf, the Western Gulf. You can see that richness here in the Western Gulf is usually higher than in the Eastern Gulf. And then the next step is you can overlay this with the social behavior of the dolphins. Of course, we have lots of behavioral data for these dolphins. So when Manuela did this, so from the foraging needs to social structure, basically, she identified 5 different habitats, and that was based on the eDNA data. So she did not use any sort of abiotic data, but more biotic data that are probably more relevant to where the dolphins are, okay? Because, they are after food. And she had identified, 5 different habitats, and basically then could sort of, find that the animals are sort of, fairly sort of specialists to each habitat. And that was a big surprise to us. So a habitat here is kind of a prayscape. It's basically a group of fish that are found, in a certain area. And these dolphins really seem to specialize on these different prey fields. And they're extremely conservative. We thought, they were ubiquitous. They would just forage on anything that comes, in front of their face. That's not the case. They're very conservative, and they don't seem to change the sort of prey fields to which they are used to. And of course, for conservation, and I will talk about that in a second, this is going to be very, important. Okay, so basically what we find is that, we've got these, varied specialists. This is sort of, the SI here. That basically tells us sort of, how specialized individuals are. So lower SI means they are more sort of, specialized to the habitat. In the Eastern Gulf, you can find this sort of, the average value is 0.6. In the Western Gulf, it's 0.5. So animals are more specialized on the habitat here. And what I said before as well is that these dolphins are extremely conservative. So they stick to their learned dietary preferences. That was a big surprise to us because we really thought they were just, eating whatever would come. And then when we lay this, the eDNA data and the stable isotope data with the social network, we basically found that these dolphins, in overlapping and foraging habitats associate more outside foraging, so the strongest in the Western Gulf and among the females. And that, so generalists here, some females, they hold very central network positions. And basically, these are also found to be more generalist in their diet than other females. The point is here that I would like to make is that we can really nicely combine social data and environmental data to explain social structure of these dolphins. And I think this is really cool because this was a PhD project that took 3 or 4 years. And that means we can reproduce this probably at the other sites to get a very good idea of, the environmental heterogeneity between the sites by looking at, remote sensing, looking at eDNA, and so on and so forth, and combining it with the behavioral data. So, and hopefully that will allow us then to compare all of these different populations so that we can hopefully, when I give this talk here again in 10 years' time, and you all will be here, tell you actually what are the environmental factors that drive social complexity in these dolphins. Okay, so I can just go through it also given the time a little bit. Basically we hope to achieve quite a lot of things. And again, stay tuned, something will come out. But the point is really that, by this comparative approach, we can for the first time really see what are the environmental factors that lead to social complexity. And that's a question that's really important for evolutionary biologists, also for us humans. The question is, why are we so complex the way we are? What role did the ecology or the environment play in this? And I think here in Western Australia, with these dolphins, we have the first time the ability to do this, in a marine organism as well. Okay, and that brings me to my last point. I also want to talk about climate change a bit. I think some of you were doing climate change research here. Who is it? Is it you guys? Can I ask what are you looking at?
Right now we're looking at intertidal levels, so high versus low, and the effect of temperature on our heart rate.
Okay. So it's temperature related, and of course there is a big problem, with marine warming and marine heat waves. Can organisms cope with that? And that's really a big question that we ask ourselves in Shark Bay as well, because we have these massive problems there as well. Shark Bay, World Heritage Area, UNESCO field site, okay? Problem is, as we can see here, these marine heat waves, they basically happening all over the place. This is, of course, El Nino here. I want to sort of, point your attention here to the west coast of Australia. In 2011, we had this massive marine heat wave where the temperatures in summer were 3 to 5 degrees above the long-term average for long-term, for a long time. And you can see, obviously, you have this massive body of water here, just the energy that must have gotten into this to warm up the water to that level. And Shark Bay, remember, Shark Bay is situated over here, was very badly affected by this heat wave. In the heat wave in 2011, roughly 35% of the seagrass in Shark Bay died off. And the entire ecosystem in Shark Bay is driven by seagrass. Okay, half the size of Switzerland, 35% of the seagrass died off. And I remember when we went to Australia back to the field in 2012, you can somehow know where the seagrass patches are, and the underwater landscape. You probably get to know, the areas here a little bit as well. And we went there, and everything, the water was milky and weird. What's going on? We did not know what was going on. No one knew that actually heat wave had happened. But basically, there was a lot of sort of material, and sediment was, in the water. It looked really milky. We didn't recognize anything. And it was basically what had happened is, this is sort of, the data that show this. This is the seagrass. It's hard to see, but basically we've got the land here. So that here with the projector, I'm sorry about this. We've got the seagrass in the Eastern Gulf, the seagrass in the Western Gulf here. This was 2002. This was the extent of in 2014. So after the heat wave, and here you can see all the red areas is the seagrass that was lost during that marine heat wave. Okay, so the temperatures were, I think, in January, February, for 5 or 6 weeks, between 3 to 5 degrees too warm. So all of the seagrass, or a lot of the seagrass, died off. And then you go, of course, does this have any sort of, consequences on the organism living, in Shark Bay? And this is, and this is actually what it looks like. So we've got, the two major species here, Posidonia and Amphibolus. This was before. This is what the seagrass, meadows looked afterwards. We could really not recognize some of these areas, totally devastated. And it was a big worry, and we did not know sort of, what would happen there. So when people looked at this, in several sort of, vertebrate species, we've got dolphins here, we've got loggerhead turtles, we've got dugongs, sea cows, green turtles, we've got cormorants, and we've got sea snakes. You can see die-off, post-die-off, that the density of all of these vertebrates massively declined, significantly declined, okay? Wasn't significant, for the loggerheads, but, for all of them, it still was a decline, not significant, but, all of them were really sort of, badly affected. And of course, this was just by counting the animals at the surface. So it was very coarse what they did, okay? So it's not sophisticated analyses. But of course, we were asking ourselves, what were the effects here on the top predators? And here is when the good thing is that we have these long-term data. So we had data from pre-heat wave, and we had data from post-heat wave. So we could really run some sophisticated demographic models to see, to what extent abundance sort of, in these top-level predators dropped, and what happened in detail here. And this is what my PhD student Felix is currently working on. So this is really, brand new data. They are about, 4 to 5 weeks old. We hope we can sort of, basically publish this soon. But the point is here that we've got the adult survival probability. This is basically up to the heat wave in 2011. So this is a sort of, temperature here. You can see it's going up and down for both gulfs. We've got basically the Western Gulf here, the Eastern Gulf here. So we've got two data, for each Gulf. You can see after the heat wave, a massive drop in adult survival probability in both Gulfs, okay? This is quite strong. We also looked at calves. Calves were also affected very badly. You can see, after the heat waves, the survival probability dropped significantly. In the Western Gulf, pretty much directly. In the Eastern Gulf, it took a little while, but also sort of, was a major drop here in the Eastern Gulf. So basically, also the calves didn't do very well. Calves were dying, and of course, you know what that means for a population. When no calves are born into a population, the population will decline. So in numbers, this means, in Western Shark Bay here, this was sort of, the abundance that we had, in our study area in the Western Gulf, roughly at, 580 animals, I believe, was down, to 300 and something. So 39% drop in abundance in the Western Gulf. And that was really interesting because we remember that, we did not see all these animals after a while. We go back every year and we take photos every time of all the individuals. And we were asking, what happened to this guy? What happened to this guy? And we haven't seen this guy in a couple of years. We haven't seen this guy. So it really seems the animals died. We can pretty much rule out that they moved somewhere else. So it really seems mortality was extremely high in the Western Gulf and also sort of, in the Eastern Gulf. There was a slight delay. Basically here, sort of, the abundance was high, then it dropped right there as well. And then, of course, that led us sort of, to the final question, also sort of one of the final questions for the talk. So thanks for bearing with me. What do these marine heat waves sort of, do in the future? Okay, so marine heat waves, we know they have a significant impact on these top predators. Dolphins in Shark Bay died because of the heat wave. The population is, maybe I should show this very quickly, kind of not really, if you look at the abundance, it's not really growing, is it? We're 2023, there seems to be sort of a slight upward trend in the Western Gulf, but we're not anywhere near the levels where it was before. And here in the Eastern Gulf, it's actually still declining. So we don't even sort of see the valley and things might not, improve. And of course, remember what I said at the beginning, long life history, calf every 4 to 5 years. So, yeah, the population might come back eventually. But if another marine heat wave hits and it goes back down again, you see what happens. It goes but up, down, down, down. And over the time, it can really sort of, have devastating consequences, on this population. So what is really important again here, only long-term data can discover these trends. Yeah, I think this is really important. We have the long-term data. If we had not been there for 40 years, we wouldn't know. Okay, so this is why these long-term data sets, and you tell the world about this, we need to fund long-term science, long-term projects. The Killer Whale project here in BC is one of those amazing projects. They have long-term data. They can look at these trends. Whatever you guys are going to do, long-term data is so, so important. And we need to tell the public about this, 2-year project, not good enough for these sort of long-lived individuals. Okay, so what then Svenja did for last part of her PhD, we also wanted to see what actually happens in the future. And this is a really cool, and I'm almost done, I know a little bit over time, but this is really amazing as well. We can actually project what the environment does in the future, and then we can look at the genetic makeup of current populations and then try to estimate whether the current genetic makeup of populations allows them to adapt to changed environments in the future. Okay, and this is what we can do with genomics. If we have, the odd genetic marker, we can do this. But now with having full genomes, and producing these full genomes on a daily scale, we can do that very quickly. When I was a young student, I was sort of your age, second year or third year. My professor said at the time at the University of Bonn in Germany, he said, it will never be possible to sequence the human genome. So never believe a professor, that's the story here. And of course, at the time we were in the lab, we did some sequencing reactions and we did 100 base pairs. And it was a great day. So we go to the pub and drink, 100 base pairs, but the human genome was 3.2 billion base pairs. So you could imagine with the technology at the time, 100 base pairs versus 3.2 billion, I think he was right. But then of course, things really improved. And now we do genomes on a daily basis. So Svenja, for her PhD, she sequenced 160 dolphin genomes, and this is feasible and not a big problem. So with these new analyses, we can actually see, can dolphins adapt to changing environments? And so basically, this is some of the different scenarios, depending on how much global warming, we sort of allow for the future. This is sort of, the pessimistic scenario here. I think we're probably pretty much there. We probably have to sort of, think about the most pessimistic scenario. And here you can see basically how much the environments, will change around in Australian waters in 100 years, given, the most, I would probably say now the most realistic scenario of, climate change over the next 100 years. So basically the darker red in area is the more environmental change we find in these waters around Australia. So there are some hotspots here, off the coast of New South Wales, here sort of, in the tropics, of Darwin as well. So remember, we have all samples from all of these populations. So we did the genomic analysis here, and then basically what we can do is a so-called genomic offset analysis. We can basically, and that basically just tells us what is the adaptability of a population, to future climate change. And the higher the genomic offset is, the worse the potential adaptability of a population is. And we can see, depending, on what markers, genetic markers we use, we can see that some areas here, basically the ones here in the southeast, but also the tropical ones here up north, they seem to be, might have very, problems to adapt to very rapid environmental change with their current genetic makeup. Whether this is going to be the case, we just don't know. Of course, this is also projecting and forecasting, and there are, error messages. So there's sort of not error messages, but, potential errors associated with it. But the point is sort of, that it seems that, some of these populations might be under more stress, in 100 years if global change sort of, and climate change proceeds as it does than other populations as well. Okay, so and basically that brings me to the end of my talk. So thank you very much for for listening. So in my group, we basically look at all of these different techniques, network analyses, genetics, sampling. We also look at drones to look at body condition, movement ecology. We do acoustics. We look at epigenetic drivers of behavior. We look at GIS habitat mapping, eDNA habitat characterization, and we also do genomic analyses. And the idea is really just to use this as tools to understand the ecological drivers of social complexity, and also, of course, look at climate change in Indo-Pacific bottlenose dolphin populations. And with that, I don't know whether we have sound or not. Just a few photos from the site, to basically finish you off, if I can say that. So that's the Indian Ocean, but we don't work there, it's just too rough for us, we don't like rough water. Osprey nest, and then the red color is of course iron oxide in the soil. You've probably seen this sort of in Australia. So the land is actually very arid. It's just really the water. Southern right whale, mum calf, pretty cool. They come back, they were pretty much close to expectation. Here we've got the arid parts as well. The land's very arid, not much, and the water, of course, is teeming with life. And as you can see, I can't get enough of this red soil, so it's just amazing. Humpback whales we have as well, pretty close to the, so you can see the size difference here. And here another sort of male behavior, two males showing these synchronous displays around the female with the drones. So this actually to strengthen their own bonds. That's our team 2 or 3 years ago on the water. You can see conditions are fabulous, This is boating weather, Dolphins in the shallows. And after a long day coming home, people are tired. No one wants to look at the camera. Manu and Svenja sort of doing eDNA sampling of Guiche Point. And a dolphin with a tool, sponge here. And Emanuele just looking at, over the nice area. And this is sort of, dolphin second order alliance chasing another one. So the other one is over here. And this is the field station that we have, much smaller than BAM, and the water is just right there, so. That's the team, it's just only a small amount of people. So it's amazing, I'm very privileged that I can work with all of these super enthusiastic students, postdocs, master students, PhDs, and so on and so forth. And it's really, really fun to go back, and work there every year. These are the universities that are involved. And that's it. Thank you very much.